Thanks for coming. And one of these days we'll do these things live again. Yeah, so lightning talks. Let me get the share screen going here. I'll tell you a little bit about the research that I do in my laboratory, and I'll tell you about me a little bit. I'll be accepting a student next semester, and so maybe you'll like what you see. Let's see here. So everybody sees that okay, I presume? So we study, hold on. We study notch signaling. It is notch signaling is a mechanism whereby two cells communicate. And I'll get to that here in a second. But for now, I like to study it because I think it's really important. And so like over here, this is one reason it's important. This is what's called the choanoflagellate. And this is thought to be the last unicellular precursor to multicellular animals, and sponges are the first multicellular animals. So notch first appears in sponges. So somehow it's really important for multicellularity. I like that. It's very fundamental. It's very basic. It's important for blood vessel formation. These are some pictures from a zebrafish. And you can see that in this bottom picture, notch has been manipulated, and you can see all these additional blood vessels that are tucked up in here. So it's important for that. This one is really exciting. People have compared the genomes of chimpanzees and humans, and they found about 860 or so genes that are only found in humans that are not found in chimpanzees. And so those 860 genes are thought to be what makes humans humans, right? And one of them is a protein called Notch2NL, and transgenic studies have been done that show that this, when they put this into a mouse, the mice have more neurons in their brain. So it seems to be important for brain size increase. And then finally, it's important for a whole bunch of diseases. Tumor angiogenesis is just one of them. But the punchline is that notch is really important mechanism for all sorts of things at all different levels of life. So I find it quite interesting. It works like this. This is a very sort of a canonical, fundamental understanding of it. Basically, it's a way for two cells to communicate. So you have one cell up here, a second cell down here. This yellow cell, or the so-called sending cell, holds out a ligand on its cell surface. And that ligand interacts, actually over here, the ligand interacts with the receptor on the so-called receiving cell. And then, it isn't all that important what happens, but basically this piece of notch gets cut off, and it goes into the nucleus, and it becomes a transcription factor to do all these different things. And so that's the basic idea. This is like the very canonical, very fundamental biology 101 sort of understanding of what notch is.
Hey, Dr. Albig, don't mean to interrupt, but I think you're using your cursor, but we see like your presentation, like the other view where you have like your notes. I don't know if you're on two different monitors.
Oh, you see this one over here? Oh,
well, now I see it over there. Yeah, so that you can follow along. So yeah, that's.
Do you see that as a full screen or just a partial screen?
Partial screen.
Let me just see here. I'm just gonna go like this then. This is fine.
Okay, there you go.
Yeah. So right. So it's cut. It makes this transcription factor complex, right? It's very fundamental biology 101 sort of thing. But what we're doing in a laboratory is we're basically sort of expanding on this. We're learning more about notch. And so there's two projects I'll tell you about real quick. One is what I'm calling the non-canonical control of notch signaling. And the second, it has to do with dimerization of these proteins. I'll show you what I mean. So the first project, right, non-canonical control of notch. I'm gonna sort of cruise through this so you get a feel for what we do. There's not a lot of detail here. But basically, years ago, we discovered that many molecules of the extracellular matrix are able to interact with notch signaling. And so we did several things: MHEP2, EGFL7, MGP is in here somewhere. And these are all things that seem to really control notch. So that's one non-canonical sort of point of notch regulation that's not in the textbooks. And we've been investigating this for several years. Notch also interacts with a whole bunch of other signaling mechanisms. For instance, like TGF-beta signaling over here, integrin signaling. We've done a lot of work with this one. And so that's another sort of aspect of Notch that we investigate in the laboratory to try to, more fully understand this. And another one is Notch seems to be controlled by many different microenvironmental stimuli. So in cells all have their own little environment they live in. It's called the microenvironment. And all these things like hypoxia, right, sugar, hyperglycemia, or even like sheer stress, how much blood flow is going down a blood vessel. All these things seem to be able to control Notch. And again, it's the same thing. We've been studying many of these different ideas. So that's sort of one big project, is sort of expanding our understanding of how Notch works and what it is. The second project then I'll talk about is this what I'm calling heterodimerization. So first off, you have to understand that there's 4 different Notch proteins in mammals. And this is a RT-PCR experiment where if you look at these different Notch proteins, you can see that, like for instance, Notch1 is widely expressed, right? These are all different cell lines and they all seem to make Notch1. Whereas other ones like Notch4, it's only expressed in these two endothelial cell lines. Okay, so there's multiple Notch proteins. They have tissue-specific expression. Sort of hold that in your memory for a minute. The second thing is that when Notch signals, it oftentimes does it in a dimer complex like this, where this is a piece of DNA, the black line, and then you can see this complex of things. The red stuff here, that's the Notch protein. The green and the blue, those are other accessory proteins. We don't have to worry about them right now. But these 2 red ones, they actually engage in a dimer on the DNA that helps the transcriptional activity. And if you sort of look at this section right up at the top here where they're meeting, and you looked and they're really close, you'd see that there are these conserved amino acids that are sort of interlacing between these 2 subunits. And these interlacing nucleotides seem to be really important, or they are important for the ability of these things to dimerize. Okay, so these are, yeah. And then the last thing is that these amino acids, 1946, 1950, 1983, 85, these amino acids are largely conserved in most of these Notch proteins. Okay, so again, there's multiple Notch proteins. They dimerize. This is just a 1-1, Notch1 dimer, but they all have shared amino acids, and those amino acids are conserved that seem to be important for dimerization. And so that raises the basic hypothesis that if we have multiple isoforms, you can have different isoforms in the cell, in the same cell type. They're all conserved as far as how they dimerize. And so basically, we came up with this hypothesis a while ago that, can the different Notch proteins form heterodimers? They've been known to make homodimers, but the heterodimers is something different. Lots of transcriptional systems can form heterodimers. And so we've been investigating this. And so far it looks like, yes, the heterodimers do form. Notch4 is by far the best, and we don't know why yet. We have no idea what it means yet. And that's going to be a big push for the future, is to try to understand why is Notch4 so good at forming heterodimers? And then what does it do? What is the consequence of it? Okay. So sort of what we do in the laboratory, sort of everyday things in the lab, we do a lot of cell culture. Okay, I haven't done animal work for many years because we have really good basic cell culture models to study Notch. So I haven't had to, and it's expensive and it's time consuming. So I haven't done it for quite a while, but someday perhaps we will again. We do a lot of DNA manipulation, so a lot of mutagenesis and cloning experiments. We do a lot of transcriptional assays, especially this thing called luciferase assay, basic things, Western blots. We do some shear stress and microfluidics experiments. We do some mass spectrometry. So, I think these are all fairly common things. Like it's like this, my motto, right? This is what I like to do with research. I like to try to find sort of the most fundamental, what I think is the most important questions. So think Notch, right? Like I mentioned, it's involved in multicellularity, it's involved in angiogenesis, and it's involved in brain size, all these really interesting things, but we don't know how it works altogether. And so that's the idea, is to find the most fundamental and important research question we can, and then approach it with what I call bread and butter experiments that we can do really well. Things like this. Okay. So it's not, so I mean, we do, I don't know, I guess take it for what it is. That's what it is. Yeah. And then about me, Yeah, I mean, I'm just a normal dude. I like to play, and I like to work too. I do a lot of dancing with my wife. We do salsa and jive, and we have a great time doing it. I fish a lot, like a lot. These are the classes I teach in the BMO program: the biomolecules one, the advanced cell biology, and the grant writing classes. So I'll see you guys a lot over the course of the first 2 years. And then something else is recently I've gotten involved in science communication. This is a book I wrote this year, and I did it with this person here is Elise Overgaard, and she's one of our current grad students. She wanted to edit the book for me, so she became the editor. And this is my daughter. She's a student at BSU who's an art student, and so she helped me. But together we put that together. So there you go. There is my quick little spiel about what I do. Can I answer any questions for you guys?